Good morning, everyone. Today, we are going to delve into a cornerstone of bioinformatics, sequence databases. These aren't just collections of information. They are the very foundation upon which much of modern biological and medical research is built. At their most fundamental level, sequence databases are exactly what they sound like. They store vast collections of nucleic acid, that's DNA and RNA and protein sequences. Think of them as the primary repositories, the initial holding places for all that raw sequence data that's being generated around the world every single day from countless sequencing projects. The importance of these databases cannot be overstated. We are currently living through an era often referred to as the genomics revolution. The advent of high-throughput sequencing technologies has led to an unprecedented explosion of sequence data. Just a few decades ago, sequencing a single gene was a monumental task. Now, we can sequence entire genomes in a matter of hours. But what do you do with all that data? That's where these databases come in. They provide a centralized, organized location for researchers globally to both deposit their new sequence information and retrieve existing data. This centralization is critical because it enables powerful analysis like comparative genomics, comparing sequences across different species to understand evolution, gene identification, helping us pinpoint specific genes responsible for traits or diseases, and crucial phylogenetic studies, which map out the evolutionary relationships between organisms. Let's look at the first type, primary sequence databases, often called archival databases. These are the ground zero for sequence data. They directly collect and archive the raw sequence data submitted by researchers from their experiments. They serve as the initial points of deposition, meaning when a scientist sequences a new piece of DNA, RNA, or protein, they submit it to one of these primary databases first. Critically, these databases collaborate extensively to ensure global data availability and non-redundancy, meaning you won't find the exact same sequence archived in multiple places, which would lead to inefficiencies. When we talk about primary nucleotide databases, three names immediately come to mind, and they form a powerful alliance. First, we have GenBank, maintained by the National Center for Biotechnology Information, or NCBI, in the USA. It's a comprehensive public database of nucleotide sequences and their protein translations. Next, we have EMBL from the European Molecular Biology Laboratory, specifically the European Bioinformatics Institute, or EBI, in the UK. This is the European counterpart to GenBank. And finally, the DDBJ, the DNA Data Bank of Japan, housed at the National Institute of Genetics in Japan. What's vital to remember is that all three, GenBank, EMBL, and DDBJ, are key components of the International Nucleotide Sequence Database Collaboration, or INSDC. This collaboration is why, despite being geographically separate, their data is synchronized daily, ensuring that researchers worldwide have access to the same, up-to-date global collection of nucleotide sequences. Moving beyond just DNA and RNA, we also have specialized protein sequence databases. These focus, as their name suggests, specifically on protein sequences. These protein sequences are often derived from the translated nucleotide sequences in the DNA databases, but they can also come from direct protein sequencing. What makes these protein databases particularly valuable is that they frequently include rich annotations. These annotations go beyond just the sequence itself to provide crucial information about the protein's function, its structural domains, and even post-translational modifications, which are chemical changes that occur after a protein is synthesized and can dramatically alter its activity. And when it comes to protein sequence databases, one name stands out as the gold standard: UniProt, the Universal Protein Resource. UniProt is truly a comprehensive, high-quality, and freely accessible resource for all things protein sequence and functional information. UniProt is actually divided into two main sections. First, there's UniProt, a SwissProt. This is the highly curated section. Records here are manually annotated and reviewed by expert biocurators, providing incredibly high-quality, non-redundant, and detailed information. Think of it as the highly polished, meticulously fact-checked version. Then, we have UniProt TREMBL, which stands for Translated EMBL Nucleotide Sequence Data Library. This section contains a much larger number of entries. These records are automatically annotated, meaning they haven't yet undergone the rigorous manual review process of SwissProt. It serves as a vast reservoir of newly sequenced proteins, waiting for expert annotation. In a sense, these sequence databases, both for nucleotides and proteins, are not just passive archives. They are dynamic, constantly growing resources that empower scientists globally to understand the very building blocks of life, driving forward research in fields from medicine to agriculture. Thank you.